in lab animals, in livestock, in plants, and environmental transformation products. It allows for the visualization of both the parent chemical and the metabolites displayed as these pathways or metabolic maps. It enables efficient and systematic comparison of metabolites across chemicals. You can compare it across species, environmental media, to identify those metabolites of potential risk and concern. Some of the key benefits of MetaPath is that it provides a structure-based searching and identification of structurally similar or unique metabolites. You can identify common metabolites as residues in plants, for instance, and see if those also occur in livestock. Provides a platform for global joint reviews and a work-share effort. And it's a systematic compilation of metabolism information that is also helping to facilitate our development of metabolism simulators. So MetaPath is recording measured data, and it has a variety of analysis tools, but it is not a predictive program. But from the systematic collection of maps into MetaPath, we are also then working on the building of metabolism simulators or predictors. So there are really three components, associated components of MetaPath. One is the MetaPath itself, the database and the data evaluation tools. But as we were developing it, we also developed what we're calling DER composers. And these are templates for efficient entry of data summaries into MetaPath. It also allows the creation of what OPP uses as data evaluation records to record what the risk assessors' evaluations were of the studies that were submitted. And it also allows auto-population of data into MetaPath, which enhances quality control through electronic data handling. And I'll demonstrate this after I talk about MetaPath a little bit so you can understand what we're actually talking about there in the composers. And again, I talked a little bit about the metabolism simulator that we are in the process of building, using, again, these observed metabolic maps, this rich data source. So this next slide just gives you an idea kind of the timeline that we're on for building of MetaPath. Right now it is mostly rat data. We knew that there was, working with OPP, that most of the data there was more information across more chemicals if you looked at the rat metabolism data. We're now starting to populate MetaPath more with livestock and plant residue data. We're also then moving towards getting also information on environmental degradates into the database. And as I talked about, we're then developing metabolism simulators to predict in areas where we don't have measured data, but building off of the measured data so that we're predicting, we understand what the domain of information is that we have so that where we predict is much more reliable. So again, MetaPath really has two components, the database, which are pathways as well as metadata, and the tools. And I'll give you examples of both of these. But to make sure you understand that some of our definitions are, I interchangeably use map and pathway, and within the database it's often called a tree, so it might show up on some of the slides. And some of the experimental matrices we're talking about are metabolites that have been measured in urine, bile, feces, and plasma. So finally, we get to what the actual MetaPath database. This is the initial display screen that appears when you open up MetaPath. On the left-hand side, there's a list of pesticides that are in the database, and they can be searched by common name, chemical name, the CAST, the agency pesticide chemical code. And in the middle, you'll see the metabolic map. So you have the parent compound at the top, the first line metabolites, or the daughters, the granddaughters, the next line, and also the next line. So it's the parent chemical and metabolites are displayed as structures. You can change your display of the actual names of the metabolites if you have names, or if they're, in this case, codes that a registrant might have submitted that and named the metabolite by a certain code. At the top is a reference, and in this case we're just showing CAS number and the parent chemical name, and then it's a rat map, but you can change this display to have a literature reference there if the data is from the literature, etc. And then there's a series of tabs at the top that you would click on to view the supporting information, the results tables, and I'll show you an example of that in the next slide here. So this is associated bioassay information that was also collected at the time the study was done for the given chemical map. And in this case, it's displayed as a results table to facilitate the interpretation by the risk assessors. And it can also be selected for export and printing. But it was important, as we worked with OPP, they said, you know, it would be really helpful. We don't necessarily need to be able to search all these tables, but we need to, instead of going back to the original study, be able to look up some of this information. And then we worked with them and identified which tables of information they thought were especially important. So as we code up information on what the metabolites were, we also code up information here as to quantity in this case, in this particular table. This next slide is another table of associated data that shows that pharmacokinetic information can also be loaded up into the database. So you have half-life, area under the curve, etc. Again, associated information that was measured in the study. We also code in data that helps you analyze the treatment group effects. It's called, within the database, it's called highlighting the treatment group. Again, it indicates which metabolites were observed under what experimental conditions. In this example, we've clicked on, it's probably a little hard to see in that pop-up box, we've clicked on the red, green, blue, and kind of a brown or a purplish color at the bottom. And so we can look at what metabolites occurred in the male, in the feces at a certain dose level versus in the urine of the males, or what showed up in the females in the different excreta. And so what you can determine is that there are certain metabolites that show up across all of the treatment groups, but there are some distinct metabolites that only show in certain treatment groups. So, for example, this metabolite here that the arrow's going to was observed only in the urine of male rats at the low dose and was not observed at the other dose levels. So it gives you a way of really thinking about analyzing the data for species and for treatment group. Another way you can analyze the data is by doing map comparisons. So one selected map can be compared to another, in this case a species comparison, or you can compare it to all the other maps in the database. Example here, metabolites were common to both maps are shown in green. The map on the left is a rat map. If you look at the top, it says species rat, on the right, species chicken. So all of the parent chemical and the three metabolites shown on the right also occur in the rat, but there are a lot of additional metabolites that were characterized in the rat, some of these being phase 2 metabolites, I believe. So it just demonstrates that you can easily do species comparisons too. So another thing that you can do to leverage this existing knowledge and looking at the chemical properties is to be able to search and group chemicals by shared features. In this case, we're doing structural searches. So what you can do, I'm showing here, we can set up a query within the database, and it can be searched by building a particular query. In this case, it's a triazole that we're looking for, the 1,2,4-triazole. And it was also set up, you can select, it was set up to look within only rat maps and oral dosing and looking for an exact match. So setting up a query like this, and once they're set up, you can store them and use them again. But this particular query will look for the occurrence of the triazole as either a parent or a metabolite across all the maps that are in the database. So the search results, in the next slide here, is within the entire database, 3 metabolites were found matching the search criteria. Triazole metabolite, found in rats, dosed by oral gavage. So then in this pop-up screen, the information is provided to trace back to the parent chemical and, in fact, that entire metabolic map. So you're really searching within a certain species as it's been set up here, but trying to find it across a whole variety of chemicals that are in the database. So you can see that if that's a particular metabolite of concern, you can ask questions about where have we seen this before, in what kind of, associated with what kind of chemicals. This just demonstrates that information can be output as Excel files from MetaPath with a lot of the various information that can be outputted. And then you can then use the information of these metabolites that were found here to load into another database like it within the OECD toolbox where you could do you could do toxicity profiling, for instance. So that's an introduction to what's in the MetaPath database. But I also want to talk a little bit about how, again, the DER composers, our system for auto-populating MetaPath. These were developed around 2 guideline studies, the pharmacokinetic metabolism and nature of the residue harmonized guidelines across, these are OECD harmonized guidelines and NAFTA harmonized guidelines. So again, it's the rat metabolism study and the nature of the residue in animals were the first 2 that we had done. And we're now expanding that to plants, nature of the residue in plants, as well as then we're going to also look at environmental degradants. So the data is entered in these composers, and the reason we did this was for a couple reasons. One was to capture the information easily in an XML file and import it right into MetaPath, and I'll show you an example of that, but also provides this text file that OPP uses as their data evaluation record. And so this will allow us to populate, the agency to auto-populate MetaPath in the future as new data is being submitted to the agency. And that will be kept as confidential information until the pesticide would be registered, and then the data will, we're working on processes to think about the open release of the data. So it's efficient, standardized data entry. It facilitates electronic data submission and electronic population, again, of MetaPath into the future. This is an example of a screenshot from the DER Composer software where we have general information on the EPA reviewers and on the chemical. The next one shows that Materials and Methods tab where we can enter in structural information. We can look at the labeled pesticide. You can use smile strings or draw the structure with a drawing package in here. The next one looks at study design and methods. This Materials and Methods captures the information in a table format similar to what had been found already in the DER, but it turned out really systemized this a lot and helps in data entry. And it's now being used, being tested by OPP contractors as data is coming into the agency. Analytical methods information is also captured because it's important to what metabolites are detected depends on what kind of methods you are using to detect them. So that was an important feature to also capture. This illustrates that tables are also built for pharmacokinetic as well as metabolite characterization data and information. There are text boxes that include the text of what the investigators' conclusions, the reviewers' comments, any deficiencies that might have been found in the study. The only thing extra that we had added to what these data evaluation records that already exist in OPP is we added the appendix tables. So one of them was to really systematically capture the treatment groups. It was information that was in these data evaluation records, but it wasn't as systematically captured. And this allows us to then have this highlight treatment group feature in MetaPath, which again, provides a lot of information. And then the last appendix table is actually the meat of how MetaPath works in that the chemical structures are input as SMILE strings, and the association of parent and metabolites is captured. This next screen on the left, it just illustrates the DER Composer data entry screen that I just talked about. And you also press a button when you're done with this, and you can have this output as a text file or a doc file. And that shows that same screen, the first page of the doc file, that's the information in that screen. And then if you look at what's circled in red, this is the reference, how it shows in the doc file. But then also how that information is now, this is a screen from within MetaPath that shows that information being captured. Another illustration are the appendix tables that I talked about in the Composer. Again, it's systematic capture of the treatment groups to auto-populate the DER. So we have male, female, dose level, duration, excretion routes, and then the information on the metabolites. This appears in another, it pre-populates these rows in another part of the DER Composer. So that's captured automatically and reduces errors by doing that. And then by placing information on the metabolites that are found at certain treatment levels, then that highlights that treatment within the database versus where there are blanks left. So just to illustrate then, this is, again, allows, by systematically capturing the data that way, allows you to have the ability to analyze what metabolites show with what treatment groups. So the benefits of the DER Composer is that it improves efficiency in the data summary and submission, maximizes the electronic data transfer and minimizing data errors that way, facilitates quality assurance of the data. We have all kinds of guidance and routines on the quality assurance. And it will help us really auto-populate agency knowledge bases to improve the workflow for both the agency as well as people submitting data. So it's getting in the hands of the risk assessors more efficiently so that they spend less time compiling the data and more time on being able to actually do the data assessments. So we also, as I mentioned, we have an OECD working group on pesticides, or under the OECD working group on pesticides, there's a MetaPath users group. So we're working with the OECD Secretariat towards the use of having these composers, or we could call them data summary builders, is what they are for the people that are submitting data. So we're trying to get these more standardly used for the rat data, livestock data, plant data again. So it'll help populate the system into the future. And using the same approach, EPA is developing these composers to facilitate electronic data capture of information submitted for the EDSP, the Endocrine Disruptor Screening Program, the Tier 1 screening data. So we're using the same kind of composers to automatically grab data so that it can be used in multiple places, stored as an XML, loaded into databases, as well as text files to evaluate the data. This next slide shows a workflow. Basically, within OPP, we worked a lot with the Residue of Concern Knowledge Base Subcommittee, or the ROCS as they call it, which is a committee that helps in the assessment of this data within the OPP risk assessment. So as data comes in, hopefully someday directly from the registrant as the XML through a DER Composer. But at this time, the contractors are developing, are using the composer and developing. Then they capture the data as an XML. The XML is then loaded into MetaPath along with the archive data that we've been able to populate into the system so far. And at the same time, then the risk assessment team within OPP gets the doc file, starts working on their assessment, but then also can pull up MetaPath and use it to do their data analysis. And everyone, again, has access to the same kind of information within that. And then as the risk assessment team submits their assessment that's reviewed by the ROC's hazard assessors, again, those people can all be using the same tool, and they can understand where the information and how the analysis was done and where the information comes from. And then what had been a draft DER can be finalized and records the actual assessment that was done and the rationale for it. So I just want to give a really quick example of MetaPath use. So you could ask a question, for instance, a new sulfonylurea, we'll call it pesticide B, is being assessed. MetaPath can be used to ask, what other sulfonylurea pesticides have we previously assessed? What types of metabolic transformations are likely to occur? What are likely the residues of concern? So we ran a search in MetaPath. Oh, I did go to, there's a little delay in that. I have to go back one. Okay, we ran a search in MetaPath for structural analogs to the parent, and the highlighted section is what we can do a substructure search on, and you can see that that same substructure resulted in, or was a component of, these 4 pesticides on the bottom. So then you can pull up the map of an individual pesticide and look at it. So this map of pesticide X shows the primary metabolic pathway, where you see ring hydroxylation, O-dealkylation, and cleavage to the sulfonamide. And where you can highlight a residue of concern, then you can look on another pesticide and see if those same residues show up in another one. So you can then look at this example, another one of those 4 pesticides that was brought up, and again you see ring hydroxylation, O-dealkylation, cleavage to the sulfonamide, potentially resulting in the same residues of concern. Just a quick and dirty sample to show you one way it could be used. So to kind of wrap up, so getting back to knowledge base or decision support systems components, if you think about MetaPath, so the inputs are the metabolic maps, the associated metadata specified in the DERs as the study information important to evaluations. And again, it's auto-populated into the future. MetaPath users define the substructures they're searching for. They define what map comparisons they want to make. Some of the outputs, MetaPath can list all the active ingredients containing a substructure of concern, common metabolites across hundreds of maps, residues of concern within the map. And then the decision support system, users identify residues of concern for the current assessment. Again, as a whole variety of searches, they can output that information and input it into things like the QSAR toolbox or other expert systems to evaluate then the potential toxicity of those pesticides. So again, the benefits of a system like this, there's desktop data availability with MetaPath. We have automated loading of new information into a proprietary database in OPP for comparison against their legacy data. There's a common data source that they're all using, allows new analyses and comparisons to be done within OPP and international partners. They can now use this tool, we're hoping, to facilitate the global joint reviews, and there's a lot of excitement to really move in this area. Allows new analyses and comparisons between chemicals, species, gender, dose levels, and again, standardized data gathering, analysis, and reporting. So the collaboration I was talking about, we're working towards a common international platform for both storing and evaluating the information. We have this international MetaPath users group established under OECD. We've had some of the participants are Health Canada, PMRA, the European Food Safety Authority, and various European member countries, the Australians, a couple different agencies there, as well as involvement for the OECD Secretariat. And I also want to point out that PMRA was very helpful in piloting with us the use of the DER composers for entering livestock data. That was very helpful. So this is the last slide I have, and I just again want to emphasize that this was built as a partnership, very importantly, working with OPP all along the way. 2 different ORD labs, both Duluth and Athens, the cooperative agreement with Ovanis McKenyon's group, working again, OPP, the ROCS committee, as well as other people in HED and EFED, and again, PMRA in Canada. So that's all I have. Thank you.
Thank you, Patricia, for this presentation. I'm going to go ahead and unmute everybody. Okay. So we have about 15 minutes for questions. If anyone has a question, just go ahead and speak up.
Hi, this is William Eck from the Army Public Health Command. I came in just a couple slides late on the introduction, and could you? This database resides on a server at EPA, so you can access it from externally.
At this point, no. It's within EPA. But because we have this international collaboration, we're working with the OECD Secretariat, there's actually a version of MetaPath that will be next version of the OECD toolbox, and I think that's, don't know if it's in the October version. That won't have necessarily all of the same data evaluation tools I showed you here. But what we're working towards is populating the database with more information within EPA, trying to use it with global partners, and then as pesticide authorities approve it, to eventually release the information, and it will be in the OECD toolbox.
Okay. One other question, please. And so right now the database is only focused on pesticides and pesticide residues?
The version used within EPA, that is correct. What I didn't mention is I think when they put the version out on the toolbox, the first early version, that was loaded with literature data. So I think it's a variety of in vitro and in vivo data for chemicals other than pesticides are also included, just what they could find at the time in searching the literature.
Okay, thank you very much.
Sure.
And I'll just jump in. Does everybody know how to access the OECD QSAR toolbox? It's actually, you can find it on the web at www.qsartoolbox.org. And like Pat was saying, I think the next version will be coming out in October. We also have a question from the chat from Cheryl Beauvais, and it says, how does MetaPath handle missing data?
Right now, what MetaPath is doing is reporting the maps as they were recorded by the registrant, with the exception of, and I'll go back here and in this slide, you can see where we have EXJ written. That stands for expert judgment. That means that in order to get, I guess, if you look at 2.1, to go from metabolite 2 to 2.11 had to have gone through there. So we put that in there. It was not something that was recorded by within the study. They either could be because it was a transient intermediate, or the detection method couldn't record that particular one, but it is immediate that should be there. So this is how we handle missing data in that respect. the metabolism simulators that we're developing from this kind of data will also help for chemicals that we don't have metabolic maps on to try to predict what those maps might be, but also be able to predict there.
Pat?
Yes.
Oh, sorry. Okay, so we have another question from the chat. It's a question about LASA Limited's technology, Meteor. Are you familiar with that Meteor technology, Pat? Can you comment on whether what may be the differences and similarities of MetaPath to the Meteor technology?
You know, I can't really comment on that. I know of it, but I really do not know much about it.
Okay. Any other questions from the phone? Okay. Well, thank you again, both Vicki. Oh, we have another question from Sheila Rao. The question is, can we just use this for evaluation only? And I think maybe that's getting at the prediction component that was mentioned, which I believe is still under development.
Well, I think it's a great research tool even without the simulators, because you can start trying to look across chemicals. You can raise hypotheses and ask them in the system already. You can try to say, well, now we see this species did have this metabolite and this one didn't. Why would that be? You know, one of the restrictions, like any system, is we need to get more data loaded into it. And that's really where we are right now, is we're really trying to populate it. But I think, yes, it can be used for all kinds of generating hypotheses that you could, as a toxicologist, go into the lab and really try to explore. I think it would be a good tool for that.
Great. Well, again, thank you, everybody, for participating. Thank you, Vicki and Pat, for presenting this information to us. And if anyone has any follow-up questions, I can try to relay them by email.
Okay.
You will be able also, as I said, to find a recording of the webinar, a link, which I'll send to the participants, but will also be on the ASCCT website. So if we don't have any more questions or anything, I guess this concludes the webinar.
Okay. Thank you, Christy.
Thank you. Yes, thank you very much.
Christy.